Good evening, friends.
Please.
Welcome to the Tuesday Critical Care and Emergency Academic Program of Yasoda Group of Hospitals. We try to bring some interesting interactions in every week, Tuesday evening. So today we have an interesting topic to interact. I think for the last couple of months, there has been a discussion on can we mix and match. So that mix and match concept came because of various reasons. The vaccine availability, the side effects, the concerns, many things. I think there were many doubts even in the clinician's mind that whenever they ask what is next, should I take the same vaccine? I think many of our clinicians also has chosen to mix and match with the available vaccines, Covaxin and Covishield also. So today we have Dr. Karthik Munta, who is our senior consultant in critical care medicine in Yasoda Somajiguda Hospital. So he will take us through the updates right now available on this heterologous vaccine regimens in COVID. Can we mix and match? Is that the way forward in the future? So over to you, Dr. Karthik.
Thank you, Dr. Venkat, sir. Good evening to all the attendees, and it's a great pleasure. Every Tuesday, we bring forward such a great and fascinating topics for you all. And as a part of this continuous medical education program, today I'm presenting this topic for the next 30 minutes on the updates on the heterologous COVID vaccine regimens, which are there as on today. The evidences are scarce, but I'll try to justify for the next 30 minutes why taking a mix of two different COVID vaccines would prove beneficial, or what are the complications or risks that or reactogenicity profile that you would face if you do so. So I would like to thank Yashoda management, Dr. Pawan and Dr. Venkat for giving me this opportunity to speak on this topic. So let us get into the topic. Can I have the slides, please? Yeah, thank you. I'm Dr. Karthik, working as Senior Consultant Intensivist at Yasoda Hospital, Somajiguda. And today's topic is fascinating one. Should we mix COVID vaccine or should we match the same COVID vaccine? As far as India is concerned, and also many countries, they have been matching the COVID vaccine. To say matching means the homologous approach of vaccines. So for the benefit of the younger generation doctors, I would like to describe this homologous vaccination is when you use the prime dose and the boost dose, and both are the same vaccine manufacturing company one, then we call it a homologous vaccination. If we use two different vaccines, which are based on two different development platforms, like you say, you use the prime dose as a Covishield, whereas a boost dose with help of a Pfizer vaccine, then you call that approach as a heterologous approach. So should we be mixing the vaccines or should we match the same vaccine? That is the topic of discussion today. Next slide, please. So as far as the Indian scenario is concerned, till the upcoming December, we would have 7 different vaccines in our armamentarium. So all of us know that AstraZeneca, Covaxin, and the Sputnik vaccine is already in the market, is being administered to the Indian population. We would see Johnson & Johnson, Moderna, ZyCoV-1, and Novavax that will be coming up by December. So this topic becomes even more important for us to know whether by the time we have these vaccines, and mind you, the upcoming months are the winter months, and we know that winters are notorious for increase in the respiratory infections. So this study and this evidence will be beneficial for all to make a decision whether we have to mix the vaccines or if we have to take a third dose vaccine, which is a different topic, and I'll come in the penultimate slides of my presentation. Then this information would be handy, and we are getting this information beforehand, before the starting of the winter months. So primary immunogenic response, if you see, of these vaccines, there are vector-derived platforms wherein for example, the AstraZeneca vaccine. This is based on an adenovirus vaccine, which is prevalent in chimpanzees, and we are using that as a vector for delivering the spike protein genome. So these vaccines, they basically, beg your pardon, this is a slight mistake in my slide. I apologize. This is a primary immunogenic effect comes from a T cell mediated immunity response in these vaccines. So these adenovirus infects the cells, and the T cell mediated immunity helps in mounting an immunity response as well as a T cell response. When you come to the mRNA vaccines, like the Pfizer and Moderna, they're most basically B cell mediated technology, wherein the B cell humoral immunity is the predominant form wherein the immunity is generated, and they attack the virus when it enters the human body and thereby give us an immunity. So if we mix these two different platforms, one a vector derived platform of a vaccine and another one and an mRNA derived platform of a vaccine, then you would get the best of both types of immunity, the T cell and versus B cell immunity. Well, this is for the theoretical discussion. What evidence do we have to say this? The next slide, please. So the necessity of mix and match, right? So what are the benefits that we get by mixing vaccine? We get a stronger immune response. That is what we discussed in the previous slide, the B cell and the T cell immunity response. And all of us know that the shortage of vaccine is a universal problem. And the flexibility of vaccine, if we have in a scarcity of a vaccine supplies, makes this kind of approach even more interesting and even more resourceful for a developing country like ours, where we are having some issues with the supply of the vaccines. And the Delta virus variants and the other variants that are coming up need a better immunogenic response. And all these vaccines give a good immunity for the variants that are emerging. But if we have a good immunity just before we are attacked by these viruses, it would be even more better for us to tackle these infections. Next slide, please. So when did this issue start, and how did this issue start of the concept of mix and match vaccines? Needless to say, these vaccines mixing have been existent before the COVID era, when we were mixing matching vaccines for Ebola infections. And in India, our rotavirus vaccination schedule also takes into account the monovalent and pentavalent vaccines. So this is not a new concept, but when we had this issue of AstraZeneca vaccine, which was banned in European Union countries, wherein they found thrombosis as a complication with the first dose of the Covishield vaccine, many countries jumped into suspending the usage of AstraZeneca vaccine over the possible side effects that were reported. And when this problem spread over all these countries, then there was suspension of this usage of AstraZeneca vaccines to make the possible even worse. Can we go to the next slide? Yeah. And when we have the Angela Merkel, Chancellor of Germany, and the Prime Minister of Italy, Mario Draghi, and taking the second dose of a different manufacturer from a different manufacturer, then the issue really came into the spotlight. And the citizens of these European Union countries started to think what would be their second dose. Mind you, all of them had taken the first dose of the vaccine, which was AstraZeneca. And now the government had increased the span of the second dose, or the duration of the second dose. And they were interested in trying out a different vaccine, which was available, that was a Pfizer or a Moderna vaccine at that point of time. And this made the scientists take up this project of mixing COVID vaccines and seeing whether it was really generating a better immunogenicity, a better reactivity with these vaccines or not. Next slide, please. So the very first study comes from Spain. Spain was one of the first earliest countries which had blocked the usage of this AstraZeneca vaccine. And this was an interesting study wherein they had studied nearly 676 subjects. And they selected the subjects who were inoculated with the prime dose of AstraZeneca vaccine and have elapsed at least 8 to 12 weeks from the first dose of vaccine. Such patients were enrolled into this study, and they made two groups, wherein the control group did not receive a second dose of AstraZeneca. They just continued with the placebo, and the intervention group, wherein they received the Pfizer-BioNTech vaccine. And then they studied the immunogenicity of that study. The next slide, please. to the surprise of ours, the data was very, very promising and even more surprising. Now, if you see at the immunogenicity data relating to COVID vaccine, the B cell immunity response and the T cell immunity response can be qualitatively assessed by these assays. So we have three kinds of antibody tests that we do for the B cell immunity. One is the receptor binding domain antibodies and the neutralizing antibody assay, whereas for the T and the spike protein assay, these are the three humoral immunity response assays that we use for quantifying the B cell immunity response. Whereas for the T cell immunity response, we have the gamma interferon assay, right? So when these three assays were compared in the control and the intervention group, if I can take your attention to this is a receptor binding assay that I'm trying to say. In the intervention group, if you see, after 14 days of this boost dose of the Pfizer BioNTech vaccine, the assay increased to 48 times. And by the 14 days, the assay increased to 77 times from the baseline. So this was a very, very promising result. And when you come to the trimeric spike protein assay, it was 22 times 7 days post the boost vaccine and 37 times post the 14 days duration. It was 37 times the baseline. So the B cell immunity response was very good. And when you come to the T cell assay, T cell immunity response, then you could see it was 45 times the baseline from the control group. So these were all very promising and very helpful data that came out of this COVID study from the Spain, which was called the CombiVac study, which was carried out in Spain. The next slide, please. Coming to the reactogenicity data and the side effects, there were much comparable side effects when compared with the homologous vaccination schedule, and there are no significant increase in the side effects when you mix the vaccine between the two different platforms. The next slide. Now, there was a major flaw in this study. They used a group where they used 2 doses of vaccine, and the duration of the boost dose was 12 weeks. And all of us know from the existing data from the AstraZeneca group that if we increase the duration of the dose between the prime dose and the boost dose, the immunity response is far, far better. And the control group did not have a second dose. So are we comparing apples and oranges here? Well, I guess so. If we have to see the data that came out of the AstraZeneca studies, which were done earlier for the efficacy, you could see that in those studies, at 28 days time, the homologous AstraZeneca vaccination showed a spike protein of 10 times the control. Whereas when you compare it with the BioNTech vaccine with the AstraZeneca, it was 37 times. And when you compare the homologous Pfizer group, and you compare it with the homologous, you compare it with the mix and match group, then the response was only 10.5 times, whereas you had 37 times in relation to the receptor binding domain. In Moderna group, it was only 6 times. And the neutralizing antibodies was only 15 times. So you can see that there is increase in the B cell immunity response when you compare it with the homologous study. But this was not a perfect study to answer the comparison between a homologous and a heterologous. It only said that if you add a second heterologous vaccination as a boost dose, the results are definitely good. Now, to address the very specific issue, the second study had been taken up by the United Kingdom group. Can I go to this next slide, please? Yeah. So this was the study which emerged from the British group. That is the Covishield group, the AstraZeneca group, which had carried out this study, wherein they had used the AstraZeneca group patients, and they have used the boost dose, which was the BioNTech vaccine. Now, if you see this study, there are 4 groups here. The first group is the homologous AstraZeneca group. The second group is the homologous, the heterologous group, wherein the AstraZeneca and the Pfizer BioNTech as a boost dose were given. The third group was homologous Pfizer BioNTech group. And the fourth group was again a heterologous, where the first dose was Pfizer and the second dose was AstraZeneca. But here, the notable point is the difference between the prime dose and the boost dose was only 28 days. But even though the results of the immunogenicity data, if you see, was very, very promising when compared to the homologous group. The response of the humoral immunity was 9.2 times the baseline when you compared with the AstraZeneca and BioNTech heterologous group in comparison to the homologous group. Now, when you come to the neutralizing antibody, it was 8.5 times, and the cellular response was almost 3.8 times more than the homologous group, which had given both doses of AstraZeneca alone. Now, when you come to the Pfizer-BioNTech group, the results are also amplifying, better than the homologous Pfizer group. But there is a catch point here. If you compare the heterologous group of AstraZeneca and Pfizer-BioNTech, and you compare the homologous Pfizer-BioNTech, the results are similar. So what does it say? It says that the heterologous group, if you are taking the AstraZeneca group and you combine it with the boost dose of Pfizer vaccine, the results are better, but they are not better than the homologous Pfizer group alone, right? So if someone has taken an AstraZeneca as a first dose and you are trying to take a Pfizer vaccine as a boost dose, then the results are far, far better than taking 2 doses of AstraZeneca. But this is a 28-day study. There is an other half of the study that is yet to be published, which is looked at 12 weeks gap between the prime dose and the boost dose, and we are awaiting those results. But these results are promising and they're encouraging. The next slide, please. This is a subgroup analysis. So this is a subgroup analysis where the immune response between the heterologous and the homologous prime and boost schedules were studied. And you can see that if you take the homologous AstraZeneca group and the heterologous group, the results are favoring the heterologous group. So if you see, all the results are favoring towards the heterologous group. But when you come to the, if you take the Pfizer as a prime dose and a booster dose as an AstraZeneca, the results are not that promising. But the responses, you should understand how these vaccines work. Like we have already discussed in the earlier slide, the AstraZeneca group vaccine basically depend on the T-cell immunity response. So when you add a B-cell platform to it, the humoral response are far, far better. But when you add a T-cell immunity response showing vaccine like AstraZeneca as a boost dose, then the T-cell immunity response are markedly increased after taking the second dose. Next slide, please. Now, to see for the other group of vaccines which are interest to our country, like the same group have started the second study, which is the COMCOV-2 study, wherein they have used the patients who have taken AstraZeneca as a prime dose, and they have compared it by taking a boost dose as Moderna or a Novax, which will be available in India by end of December. And the results are yet to be published. And even they have compared the same platform vaccines, like the Pfizer dose, which is a primarily an antibody dependent immunogenicity vaccine, and they have compared with another mRNA based platform vaccine, that is Moderna. The results will be interesting. We have to wait for a month, and these results are expected to come out in August. So we'll have good amount of data, and with this data, we can see whether we can really mix these vaccines or not. The next slide. Now, this is of interest to all. So most of these countries have already taken 2 doses of vaccine. Now we are interested to know whether we need to take the third booster dose or not, or our immunogenicity is good. Do we need to wait for 6 months? Do we need to wait for 10 months? What is the data that is as on today? We don't know clearly, but the emerging data comes, the earliest emerging data comes from Israel, where the health ministry had reported that the vaccine efficacy of the Pfizer vaccine had been dropping to the Delta variant, which is now prevailing in Israel. Israel was one of the first countries that have already vaccinated the entire population, and they have seen that there was increase in the illness because of the Delta strain. And this was, the efficacy was dropping from 95% to 64%. And the country had already said started murmuring about the use of the third dose. And when the study that was carried by Israel was published, then this data was used by the Pfizer group to approach the CDC for clearance for the third booster dose. The next slide, please. So the CDC is yet to decide and give its nod for usage of the third booster dose. As of now, for the Pfizer, it is yet to be decided in the interest of the public. But some countries have already tried and started giving the booster dose, like Thailand and Bahrain, where some of the countries which have come up with already administering booster doses. Bahrain was one country which was given the Sinovac or the Chinese vaccine in the earlier days, and the results or the antibody response was not significant after 6 months, and they have started administering the third booster dose in their country. Next slide, please. Can I have the next slide, please? Hello? Can I go a slide back?
Slides are changing. Karthik, slides are changing. Not able to see.
Can you see, sir?
No, sir. The previous slide. Right. So yeah. So this, can you see the slide, sir?
Yeah.
Sorry for the interruption. We have 3 more slides to go. I'll be completing my talk. Yeah, we'll take from here. Yeah. So we have another strain of, can I make it a full screen, please? Yeah. Yeah, thank you. So the AstraZeneca is already into the producing a variant vaccine, what it had already produced. One thing we have to remember with the AstraZeneca vaccine, that it uses a vector, which is adenovirus. So when we have introduced this virus into the body, the antibodies are also generated for the vector virus. So when we again use the same vector virus, there is going to be some drop in the immunity. So the AstraZeneca has also incorporated the changes and also tweaked the spike protein genome, and is already under the second or third phase trials to give a new variant vaccine. It's already under the pipeline. So we have to wait for this vaccine in order to take a boost dose. If we can wait, we'll have much more information about this vaccine. Next slide, please. So what are the real possibilities in India? All of us are interested regarding this. We have Covaxin, half of our population, which is inoculated with Covaxin. We have AstraZeneca, which is the major predominant vaccine here. So the Covaxin has already initiated its study for the third booster dose, and the results will be out. With its interim results will come out in August, and the final results will be in November regarding whether we can take a third booster dose. AstraZeneca, has already discussed, has been tweaking its vaccine to combat its variant. And the Pfizer is in its final stages of collaboration to get nod. Moderna is already going to be marketed by Cipla. Novavax is going to be marketed soon. And indigenous mRNA Indian vaccine is also in the pipeline. So there are a couple of possibilities that can emerge in coming months. And with the evidences that are going to come from the CoviBoost ongoing study and the COMCOV2 study, we'll have much more data to say whether the third booster dose is really required. And if it is required, by what time should we be taking this booster dose? So possibly the data as on now is not convincing and not clear, and we need to wait some more time for the clarity to come. So if I have to summarize my presentation, and I would say that heterologous vaccination is promising, the concept is promising, the immunogenicity is good, and definitely there's going to be an mounting evidence towards the heterologous vaccine. The dosing interval is always going to be a vital factor. Needless to say, if it's a homologous vaccination or be it a heterologous vaccination, the dosing interval will play a prominent role. We have to wait for the data to support the usage of third booster dose to evolve, and India has to get its data regarding mix and match policy before we give a direction. So these are the most prominent points that we need to keep. And we have to also understand that all these are in vitro studies. All these are immunogenicity studies. The earlier studies for giving an emergency approval from all these vaccines came from the efficacy data. They are real life data. They are data that comes out from patients. These are not exactly the data that are coming out regarding the efficacy. If we start thinking of efficacy at this point of time, then it will take a long time for us to get the results. So we have to rely on the immunogenicity data for the time being. So with the upcoming results of COMCOV2 and COVID boost, the results will be in a better position to answer this. But definitely we can say that heterologous vaccine is no way inferior to the homologous vaccine. The immunogenicity is better. The side effect profile is acceptable. And with the coming vaccines in India, definitely these studies will throw some more light, and we can even, the scarcity issue will be resolved, and we can mix and match these vaccines to take. And this is some of the information that I could gather out of these scarce data that was available on this topic. I hope most of you had gathered some information and you have found my presentation interesting. Thank you all for your patience here.
Thank you, Karthik, for that lucid presentation. I think the overall concept is everybody wants to know about what is this mix and match vaccination, what is it heterologous regimes. These are all in the minds of many. We have some interesting questions also. Karthik, there were questions asking ki now most of our healthcare professionals initially got a Covishield as the first vaccine, and even the 1 month, in a 4 weeks time, they got the second dose. They are trying to know ki now if at all they need a third booster in some time or the other, what will be the ideal as for your combination of T cell mediation is with Covishield? What will be the alternative? Is it a Moderna, Pfizer, or Covaxin? What will be the ideal alternative third booster?
Yeah, so sir, your question has two parts in it. The first part I'll try to answer. The first part is when to take the third dose. Am I right?
Yeah, third booster. Yeah.
So all the preliminary data say that there is no urgency to take within first 6 months of taking the second dose. So I think 6 months is an acceptable time. You can wait for taking a third booster dose. Now, whether taking a third booster dose really evidence-based, is it an evidence-based that I am saying? No, their evidence is yet to come to say about that. But if the time period you see, most of the data that might come up, that is the interesting study for this aspect is the COVID boost study, which is the third study that is being taken up by the AstraZeneca group. And this study will show the light, throw the light on the immunogenicity. And if I have to say, you can wait for 6 months from the second dose and then think about taking the third dose. And what vaccine I have to take, that's the second part of your question. Now, as the data that I have shown you before, for 2-dose regimen, if you are mixing and matching, the responses are good. So going by that analogy, I would say that if you are taking a vector-based platform vaccine like your AstraZeneca, Sputnik, Johnson & Johnson, and all these vaccines, then you could combine it with an mRNA platform vaccine like Pfizer, Moderna. These would be an ideal match. And the data exists with the Pfizer as of now. And the outcomes with Pfizer when you are mixing with AstraZeneca are really promising. So I would say if I have to select one vaccine out of these, then it would be Pfizer. And what would be the time? 6 months from the second dose.
Now we are coming closer to that 6 months. If you see, the vaccination has been rolled in the first part of the January. And then if you take 6 months of the second dose, that means from February, you will land up somewhere in August and September. I think we are a month or two away from that point of 6 months.
Yes.
The second part is, so we now have a Sputnik which is available.
Yeah.
There are also concerns related to taking Covaxin. So what about, even Sputnik is a vector-borne, and again go with the T cell mediated, mixing matching with the same kind of vector-borne and same T cell mediation, will it have any help to play?
Yeah, that's an interesting question. I don't have data to answer that, but definitely the studies are going to come, at least in Indian scenario. They will be doing with 2 different vector-borne vaccines. So one Covaxin is not exactly vector-borne; it's more of a whole cell inactivated, wholesale inactivated vaccine. If you are talking about vector-borne vaccines, then Sputnik and Covishield would be an ideal choice to combine. And there have been data, and they're not unconfirmed data, say that yes, they can be mixed. But we have to see the immunogenicity. What is the immunogenic response going to be? And if you see Sputnik is slightly different from AstraZeneca. Sputnik takes two different vectors. It's not one vector. Covishield takes single vector in its manufacturing. So that's a slight difference. Whether if you combine Covishield with a Sputnik, what would be the immunogenicity? Probably yes, it's a good proposition to start off investigating on that. And we don't have option in our country. Options in our country per se are very, very less. So we have to work within the options, and definitely the central government is looking into this, trying to prepare a study wherein the Covaxin and the Covishield are combined with the newly entering market vaccines. So we have to talk about Novavax, we have to talk about Moderna, we have to talk about Sputnik. All these combinations are being thought of. But what all data that exist as of now are from the Western data. And these data are basically mixing a T-cell response and a B-cell response. So we can talk, I can say
the Sputnik, the other add-on, I think there is a efficacy percentage. If you see whatever AstraZeneca has published their own data of efficacy, even preventing the hospitalization or the preventing the disease as a 70% or 60% in variant. Whereas Sputnik has given its own number around 92, closer to those mRNA vaccines which has been. So I think that is another point where it has a two-vector mediation and also having a higher maybe effective protection effectiveness. Is that another point where many of those who has taken the vaccine from the Covishield has their own concern. Again, the second part, as you said, Covaxin, combining Covaxin. It's not a vector borne. We know it is a killed inactivated vaccine. The whole virion is involved in this. So if you remember in the early part of this so-called variant, you say Delta variant, you say the double mutant variants, there were a lot of rumors, concerns, and even Covaxin themselves claiming that they cover all these mutant variants, whereas the spike protein mRNA may not. So this is because of it's the whole virion or a killed inactivated virus which has been implemented. And so there you saw there is a huge surge in demand of Covaxin over a Covishield. People, even in India, I still remember getting a lot of calls. Where is the Covaxin available? We want to go and take Covaxin. So what do you think of taking a booster dose with Covaxin now after Covishield, or taking a Covishield booster after a Covaxin?
Sir, this is already been practiced. Many doctors also whom I know have taken Covaxin after taking Covishield.
Yes.
Not reported any side effects with that. But to say that without evidence would it be proper? But if you combine it with the B-cell vaccine, it will be even more productive. That is what my opinion is. But in India, what B-cell platform vaccines we have, then as on today, the nearest vaccine that we are thinking about is probably going to be Novavax or Moderna, which is yet to come, which has been given the nod and approval by the center, but it's yet to be marketed. So we have to wait for those vaccines. But if your question is whether we can mix Covaxin and Covishield, yes, you could start doing that, but you need data, you need studies on that.
You are aware of any ongoing studies, anybody attempting it?
In India?
Yes.
No, sir, not yet. The central
in India only we will have this possibility because these are the two vaccines which are rampantly available and used in India.
Yeah, one thing I would like to say, this is regarding specifically you brought the topic of Covaxin. Now, Covaxin being an inactivated virus will not attack the cells. So there is no question of you getting a T-cell immunity there. At the max, you'll be getting a humoral immunity with a Covaxin. But when you come with a Covishield, you will get a T-cell immunity and thereby also get a B-cell immunity. So that could also be an ideal choice to mix a Covishield with a Covaxin. That wouldn't be a bad idea. So Covaxin and Covishield or Covaxin and Sputnik, you could try in Indian scenario. If we get some data on that, definitely that will be a promising heterologous combination. But regarding this what I'm talking about, these studies are whether it's a 2-dose study or a 3-dose study, that needs to be clear. If your question is about 3-dose study, then definitely the risk will be less, because already we have taken 2 doses of a similar vaccine and we are trying to combine a third new dose that is going to be a single dose. So probably results should be good in my understanding, but we have to wait for the data to emerge.
Yeah, other question, I think third dose would be Moderna or Covaxin. I think he already answered. He felt the T-cell mediated Moderna would be his choice, or a Pfizer vaccine, if available, will be his choice. When to start chemotherapy after vaccination? I think that's a actually out of track question, but you want to answer that question?
No idea, sir. I mean,
so I think they are not live vaccines. So we either discontinue or stop chemotherapy for that. I think it's not like any live vaccines which we actually do not administer. I think it's after the regular time period, the chemotherapy need to be continued in its own time schedule. There is no need to wait or change your schedule for just a vaccination. So I think with that, I think Karthik, we do understand at this point of time, we have a lot of theoretical assumptions, theoretical jumblings, and we have some information with the currently available 2 or 3 studies that still we can have a mix and match, and there is some theoretical basis for that. But as you said, in India right now, we still are waiting for an mRNA vaccine to be available to any of us. So if at all it will be available, that can be used as a third dose, and that may be as a second booster after your first prime, and then the first booster. Then will be the second booster as a mRNA vaccine. And coming to the other two, like using a vector-borne Sputnik or your Covaxin in combination, that I think we need studies. We need probably further analysis and data and literature supporting it. At this point of time, we do not have a clear evidence or supporting data for that, but these are all available theoretical assumption that this can be an option. And the other problem in India right now or anywhere in the world is availability. The number of vaccines or vaccine doses which are required to vaccinate a major portion of the population is huge, and that too particularly the countries of this size like India. So we need multiple companies' vaccines to come up and help us. So we right now have 3 vaccines already available, that is Covishield, Covaxin, and Sputnik. And we are waiting for some mRNA vaccines and maybe Johnson and Johnson and other vaccines. So you are saying all of them will be available by December. Is there any official announcement of that?
Yeah, when we had this second wave, the government had already approved that if you have an approval from the CDC or from the European Union or from Japan, that is what I could gather the information from the internet and from scientific literature, that they're going to give clearance for that. And they have given not for these vaccines, but still Pfizer BioNTech is under discussion. It's not yet given not for that. So probably these vaccines are going to come into the country, and we have to keep in mind that we are upcoming months, the winter is going to start. So before winter starts, if we have information on this study, specifically if we have information on the Indian scenario, Indian vaccine combinations, it will be helpful for us to decide about the third booster dose. And before I forget, I just wanted to make one more point with your permission, sir, regarding the immunogenicity. Most of us have this notion that after taking vaccine, we get the immunoglobulin levels checked. And if our immunoglobulin levels are low, then we feel probably we are not mounted a response to the vaccine that we intend to have after taking 2 doses of vaccine. This is not specifically true. What we are checking, like how we have discussed in this slide, there are 3 different kinds of humoral immunity quantitative tests that we do. The best one being the neutralizing antibody, which we cannot do in a laboratory kind of situation. They're mostly study-based tests, and the other 2 are the receptor binding domain assay and the trimeric spike protein test. So we do for the spike protein IgG, but this is only half of the story. We are not interpreting the T cell immunity response that comes along with this vaccine. And we are not seeing for the receptor binding domain assay. We are just seeing one test of humoral immunity, and we are basing our judgments only on that one test. So it is probably jumping the gun, and we need to see the entirety as a whole. So these vaccines also develop memory T cells. They also develop memory B cells. These are very, very important, and you don't have a test to quantify them. And that is why the efficacy comes into the picture. Efficacy takes into the picture the clinical outcomes of these vaccines, which we have been heavily relying upon in order to say whether the vaccines are agreeable to be given to general population at large or not. So if you see the IgG vaccine, your numbers are low, you not be depressed, or you not be dejected, and not be in a hurry to take a third booster dose, right? So whatever is the efficacy, whatever is the immunogenicity, according to WHO, all these vaccines that have been approved by WHO, all these have a good percentage of cover for those patients who are really become sick. So if you see any patient who have become sick, and if you see whether they have become sick after taking the vaccine, the sickness has not progressed. So all these vaccines which we are discussing, whatever mRNA or the vector-borne vaccines that are approved by WHO, they have been good enough, they have been effective enough in preventing the progression of the severity of the illness. That is where it is important. Getting an infection, if you see the name of Covishield, according to Adar Poonawalla, what he says is that it's a shield. It's not only going to shield you from the severity of infection, it's not going to protect you from getting an infection. If you get an infection also, you're not going to progress to the severity. So all these vaccines are good, and you not rely on a single IgG humoral response to judge the performance of your vaccine.
Yeah, adding to that point, few, as you said, ki measure their antibodies, and they, after the vaccination, and they realize ki the antibodies are not adequate, they get worried. There are other concerns. Antibodies were good and adequate after the infection or after a vaccination, and they faded away after 6 months, and they again getting worried. So as he rightly pointed out, it is not just the antibody mediation. The T cell memory plays its own important role in actually memorizing the virus. So it gives you the protection which has been seen in the previous all the other vaccines which we have been using for centuries. The other part is, see, none of the vaccines which we use for the last maybe few decades were not 100% protective. If you take chickenpox, you take other smallpox, polio, or any other vaccines, including the influenza. See, the effectiveness was always varying between 70% or 80%, and that should be quite good enough to protect you. But the problem with the current COVID-19 is its transmissibility. It is such a highly infectious disease that it is coming wave after the wave. And about the mutation, Karthik, do you want to actually comment on that? See, last I think 6 months or being a big news. So that the mutation or the mutant variant, you say whether it is a double mutant, delta plus, or many, and vaccines claiming they to be covered, and every day you get a news that this vaccine covers this, that vaccine covers that. Contrary to, see, many of us, even though claimed that this vaccines, whatever the vaccine, they protect hospitalization, they protect the severity, they protect the progression. But we do saw a few of our patients who got fully vaccinated, two vaccines, still maybe that percentage was very small, but they still got into the severe disease, and few of them lost their life. So see, all of us should still continue to protect ourselves with all this standard COVID precautions along with the vaccination, and then probably we will wait for. You want to comment on that mutant?
Yes, sir. So this is an interesting thing. Now, when I was looking through the data for the third booster dose, especially the evidence that we have comes from a single study from Israel, where they have seen that after taking 2 doses of Pfizer BioNTech vaccine, they could see that the emerging Delta virus was a pretty nuisance in their country. And especially children who were between the age groups of 14 to 16, they formed almost a bulk of nearly 60% of that population that was becoming infected. Right. So now they had to find a solution to that. So they could not tweak the genome of the mRNA vaccine that already has been produced and have been distributed. Now, what they say is when the variant emerges because of the infectivity or the severity and the numbers of infection rising, the spike protein and the spike protein binding sites, they go transformational changes. And now we cannot change the genomic sequence and we try to produce a newer antibodies. Rather than that, what they have seen when they compared with regular strains which were prevalent earlier and the Delta strain now, the attachment of these antibodies, this was basically a neutralizing antibody study that I'm saying. They have seen that the antibodies, say about one virus was attacked by 10 antibodies initially because the antibody response was good. But now, because of these variants, only 5 or 4 antibodies could attack these viruses. Now, in this time, like we have already elapsed 6 months from our second dose, the antibody quantity would have gone down. And with the Delta variants emerging and the winter months coming, definitely the number or the quantity of antibody should be at summit if we have to attack these variant mutant strains. So if we consider a third booster dose, though the antibodies are the same, when the number of the antibodies are more, then the number of antibodies that could attack these mutant variants would also be more, and the efficacy would also be better. So that is why, that is one more reason that they have been advocating the need for use of third booster dose from the study in Israel. So that is a basis that was formed for giving the third booster dose. So we could increase the number of antibodies, and thereby with the present genome that is existent, we can overcome the variant vaccines. But as I have shown in the earlier slides, the AstraZeneca has already come up with a change in its genome of manufacturing these vaccines. And definitely the next vaccine that AstraZeneca is going to be produced and probably will come out in few months will not be the same vaccine that has been used initially in the Covishield. So these are interesting things. These are evolving science that is existing for the present scenario wherein variants and mutants are coming into the picture.
Yeah, thank you, Karthik. Thank you for that elaborative, lucid, interactive session. I thank all the participants for their patience listening and being a part of it and participating. I think we will be bringing the similar kind of interactive or interesting interactive sessions in the future. With this, I thank Karthik and conclude the session. Thank you, Karthik.
Thank you. Thank you, Vanita. Thank you, sir.